Next up, we have Dr. Nabi, who has been doing amazing work on subcellular, submembrane components of the cell, which is a great, which is a great application for super resolution microscopy. So Dr. Nabi is a professor at University of British Columbia and has been doing cutting edge super res for a long time using classical methods, and now he's starting to dabble on the dark side of machine learning. And we'll go to hear more about that now. And we're good to go. Thanks.
I'd like to thank the organizers, thank Claire for organizing all this. Also thank Ethan for all the work. I'm going to talk to you a little bit, continue to share. I don't know how to get rid of that. There we go. I don't know. So, do you have any idea what's going on?
No. But let's just go ahead and try the other option.
Okay, well, I think we're super resolution microscopy generates images. I would say these images are akin to what's called the outer structure of a building. And as we can go and use super resolution, we're going to get higher magnification, we're going to get more detail on a particular part of the image. You can see here a couple of examples. This is single molecule D-storm for caveolin 1, and this is STED for the endoplasmic reticulum. The question is, how do we move past the image? How do we go and find what's really the design of the molecular design, the molecular architecture of the structure, what an architect would call the inner structure? And what we've been doing is using artificial intelligence and machine learning to develop approaches, particularly, and I'll talk to you a little bit about what we've been doing with analysis of single molecule resolution. So. Having a little trouble here. Sorry. Okay. So the work I'm going to talk to you about is work that was done in collaboration with Ghassan Hamarni, a computer scientist at Simon Fraser University. He's in Burnaby, on the east side of Vancouver. I'm at UBC on the west side. And we used to meet with my student Ray, who is here today. So it's really great to see Ray here in the audience, and also with Ghassan's student Ismail in a cafe, and we used to talk, and we had very extended conversations, really bridging this divide of the biology to the computer science. This has been 10 years ongoing, and we now have, you can see all the people have gone to the lab and interacted, and I think we really have a really robust collaboration and a really productive and really interesting and fun collaboration. So, and pursuing some interdisciplinary science. It's really interesting, it's really fun. I'm going to talk to you about SMLM. I do not need to introduce SMLM. AK has done a great job. But what I will say, and I'm going to keep my, sorry, I don't have the pointer, but all these D-storm, POM, GSD, DNA paint, these are MUNFLUX, RESI, these are all single molecule approaches. I'm sure you've heard the names. What they all do it a little bit differently, but what they all do is they generate, as Yuri said before, lots of XYZ localizations, and this can be millions. And what are XYZ localizations? XYZ localizations are a point cloud. It's a cloud of points. It's a distribution of points in 3D space. How do you analyze a point cloud? Well, fundamentally, a point cloud is a graph. The approach to analyze graphs is really network analysis and essentially connecting the dots. And the way you connect the dots is you really go and you start to see, well, which dots are in close proximity to each other. And we set the limit for proximity using a threshold called the proximity threshold. And as you can see, that as you go and you increase the proximity threshold, you start to get more and more connections. These blue lines are what we call edges, and you get more edges, and you can start to see that every red dot starts to have a number of edges, and that number, you can see for the bottom on the bottom right, 13 edges. and that's the degree. So every point gets a number that's a degree. The degree is the measure of the number of edges formed between nodes at a certain proximity threshold. And so we developed a machine learning algorithm to analyze the connections, the edges, based on network analysis. And fundamentally what this process, this was developed by Ismail Kater, a former student in Ghassan's lab. And if you want, you can just click on, go to the QR code, and you can find access to the software and the information about SuperResNet, what we call SuperResNet. Essentially, fundamentally, we load essentially a CSV file of all the localizations. We apply network analysis. We merge localizations to deal with multiple blinking issues that are prevalent in single molecule analysis. We filter the data versus a random network. We then segment the data. We identify clusters within the data. And then we go and we classify them using a whole bunch of different features, about 30 different features, and that allows us to group the data into different groups. We then have, once we have the groups, we can go and we can visualize, and we can look at individual blobs, we can look at modules of different blobs, and we can also go, and I'll show you a little bit of new data that we have on looking at interaction between two different channels and at the molecular level. And then we can output a whole bunch of quantitative output that we can use for statistical analysis. And so this is just an example. This is some MinFlux data. This is MinFlux data published and downloaded from the internet. It's from a paper from Stefan Howell's group of clathrin-coated pits. The image they provide is on the top. And what SuperResNet can do is can take those blobs, can identify the blobs, and generate different structures. Actually look at structures. We can identify structures which are more clathrin vesicles, more spherical, more clathrin pit-like, and also flat lattices. And so we're able to obtain very, very defined molecular information from the single molecule localizations of MinFlux or any other single molecule data. So the protein that we study and what started us working on single molecule was really caveolin-1. Caveolin-1, CAV1, is the code protein for caveoli. You can see the caveoli, these very nice little smooth invaginations of the plasma membrane. At the bottom, you can see a deep etch image, EM image of the caveoli, where you see the typical striated coat. Caveoli formation requires an adapter protein called cavin-1. The caveolin down there is in pink and cavin-1 in green. But the issue was, and this was an issue we dealt with, was that you have lots of data. We had a lot of data showing there were functional roles for caveolin independently of caveoli, independently of cavin-1. And this was happening in structures that we called scaffolds. The trouble was, well, how do you identify a scaffold? Okay, and a scaffold, you can go by fluorescent microscopy. In that image, caveolin is red. This is confocal. And you have a lot of dots. And how do you know which dots are scaffold, which dots are caveoli? Very difficult. By EM, you can clearly see the gold labeling of the caveoli, but the other labeling is really, really diffuse and hard to decipher. And so we set up and I'm not going to go into details, but we set up essentially a weak supervised approach to identify caveoli, comparing cells that had caveoli and cavin to cells that didn't. And we identified essentially 4 structures in the plasma membrane of these cells. We identified structures that we called S1A scaffolds of 10 to 15 CAV1s. You can see it over there. And this really matches what's known. Cryo-EM defined recently an 8S complex, this really nice disc-like structure with a barrel, and we were able to identify this by a single molecule. We also identified S1B scaffolds, which are essentially dimers, and this is work also of somebody in the audience. So Vivian is here, and Kang is here somewhere, so I'm really happy to see you guys here too. I don't normally present it with the people who have done the work in the audience, so that's pretty nice for me. And we also found higher order structures, 4 to 5, which form this hemispherical structure, as well as caveoli at the bottom. And the caveoli was really nice because when we went and we did our modularity analysis, we found that the caveoli were formed of 6 to 7 S1A dimers, which corresponded to the dodecahedral structure that was proposed by Cryo-EM and in a previous paper from the Hellenius lab. And I show here the striated structure, just because and this is what a caveoli looks like, and I'm going to come back to that later. and show you some. So we were very interested in looking at how does caveolin and cavin interact in the cell. We set up a dual channel spectral demixing D-storm using an abolite microscope that we accessed at the Institut Curie in Paris. This enables us, spectral demixing allows us to use 2 far-red probes, allows them to acquire them simultaneously. It limits really the drift issue, the chromatic aberration issue, so that we can really assess molecular interaction at the molecular scale. And so you can see here the output is that in green is cavin1 and red is cav1. You can clearly see that the large blobs are labeled for cavin1 and cav1. And you can see that when we quantify the output blob from SuperResNet, that the majority of caveoli are labeled for cavin1. Very few of our 8S complexes or S1A scaffolds are labeled for cavin1, but there are a number of S2 and S1B intermediate scaffolds that are labeled. And you can see that in the images below. In the image below, you can see that each of the classes are separately presented in red. So the caveoli in red over here, clearly labeled with cavin1 in green, while at the other end you have S1A scaffolds that are not labeled to a much lesser extent. We can go into the data and we can now analyze a whole number of properties. And I'm showing you a few examples here. We can compare the blobs, the cavin interacting blobs with the not interacting blobs. We can see that the size of the not interacting blobs is smaller, but we also see that we see changes in the actual regions of the blobs. And so we can see that the modularity, the blobs that don't interact are less modular, while those have reduced density. We see the same thing for caveoli, and we can see that caveoli change in size, and that the regions associated with the caveoli are changing. If we actually go and image this, we can actually see that the blue and red interacting zones are regions where the two fluorophores, the 2 molecules, are in close proximity, and we can see that there's a sort of a region on the edge of the caveoli where the cavin's interacting, which very much matches this Clem image of cavin1 labeling the striations instead. And so I think what we're able to do here is that we can show that cavin1 interaction with caveoli impacts caveolin structures, but also impacts local organization of cavin1 in these oligomers. And finally, I'm just going to finish by talking a little bit about super resolution, maybe bring up some topics for discussion. First, I think we're going to be moving past confocal. I think it's important to think there's lots of options for moving past confocal, hardware, but also software possibilities. I think we really want to develop high resolution, high speed, multi-channel, volumetric imaging for live cell imaging. Okay? We also have to move, if we're going to go to super resolution, we have to move past co-localization as a quantitative metric. We have to actually start measuring spatial interaction between subcellular structures. And finally, I think that these new tools, these new single molecule tools, be it hardware but also software, are really providing us the means to do in-cell structural biology. And I'd just like to thank the people who did the work, in particular Ismail, who developed the software, but also Lydia Lee in my lab, who did the work that I presented. And thanks to all the funding agencies, and thank you.